Welcome to Series 2 on Amino Acid MCQs. We'll continue our lectures on our question and answers on classification, chemistry, properties, reactions of amino acid, peptides, and the tests. So let's continue. Please do not forget to subscribe and click on the notification bell. So, which is essential? Which is essential? We have glycine, alanine, lysine, and aspartate. The answer to that question is lysine. Is lysine. So lysine is an essential amino acid among the options here. So, but now, like I said in the earlier lecture, essential amino acids are those amino acids that our body cannot produce, so we need them in our diets. Why the non-essential amino acids, those ones our body can produce, so we're not necessarily in them via our diet. So we have some of the essential amino acids. We have the phenylalanine, we have the phenylalanine, we have the leucine, we have the leucine, C-I-N-E, we have the isoleucine, we have the isoleucine, we have the methionine, we have the lysine, we have the methionine, we have the tryptophan, T-R-Y-P, tryptophan. So we have the valine. So these are essential amino acids. Essential amino acids. So we also have the non-essential, where we talk about the alanine, the asparagine, the aspartic acid, the glutamic acid, so many of them. So that will make it the 20 amino acids I found in Pro. But we also have others that we call the conditional essential amino acid. When we say conditional essential amino acid, are those amino acids that they are required, maybe in a diseased condition. So in a disease condition, we might be lacking those amino acids, where the management of that disease might require that we give the individual those essential amino acids those amino acids are not essential if the individual is healthy. So we have which is non-essential from my list on the previous slide. You know that methionine is essential. Alanine is not essential. Threonine is essential. So the answer to this question is alanine. It can be synthesized by the body. So we have the sulfur-containing amino acids. There are 2 sulfur-containing amino acids out of the 20 amino acids are found in proteins. 2 of them is cysteine and methionine. Methionine are the 2 sulfur-containing amino acids. So either one that you see on the option will be the answer. So we have methionine. If you also have cysteine here among the option, that means you choose the 2 answers. So aromatic amino acid. Which of the following is an aromatic amino acid? When we say aromatic amino acid, we mean the one that have benzene or aromatic ring. Benzene or aromatic ring. Why I use that language is histidine, which was not regarded as an aromatic amino acid before, is now regarded as an aromatic amino acid. It doesn't have a benzene ring, but it has an aromatic ring. So we'll say aromatic ring, thyroxine, phenylalanine, and tryptophan are the aromatic benzene containing aromatic amino acid. So which of them you see in the option will be the answer. So the answer to that question is thyroxine. That's an aromatic ring. So which amino acid is regarded as an amino acid. It's no longer an amino acid, it's an amino acid. It's an amino acid because the amino group on the amino acid is a secondary amino group. It's a secondary amino group. How do we have such structure? We have, let's assume we have COOH or CO minus. We have the carbon, we have the nitrogen. Now this one comes this way. So you can see that we have 1, 2, 3, 4, 5. So you can see that we have, this is a proline. This is proline. So we have, so this nitrogen here is housed, or let me put it, is in between these two carbon, this carbon and this carbon. So this nitrogen is a secondary nitrogen. So that's why we call it an amino acid. Amino acid rather than amino acid. So the answer to that question is proline. Proline is an amino acid. So peptide bond is also called an amide bond. Amide bond. The peptide bond is a bond that is formed between two amino acids. Usually, the first amino acid will contribute in the carboxyl group, while the second amino acid will contribute in the amino group. So it's a bond between the amino group and the carboxyl group that makes up the peptide bond. So we can also call it an amide bond or amide linkage. So we have a nature like this. This is the carbon that has, this is one amino acid. Sorry, this carbon would attach to the nitrogen of the second amino acid. We have OH, so we have hydrogen. So the bond between these two here is called an amide bond or a peptide bond. So the bond looks like this to nitrogen. So peptide bond formation involves, for we to form that bond, something must be removed, which is water molecule. So it's a condensation kind of reaction. It's a condensation reaction. Remember, we have COOH reacting with NH3 plus of the second amino acid. So this OH is removed, one hydrogen is removed here. So what do we have? We have C double bond oxygen to nitrogen, then two hydrogen left. Then the nitrogen is continue with the alpha carbon, which has one hydrogen and the arrow group, then the carboxyl group of this particular amino acid. This one will also have the alpha carbon attached to the amino group. We have hydrogen, we have the arrow group. So this arrow group and this arrow group tells you that there are two amino acids. Then the OH group that is left here and the one hydrogen that is left here will give us H2O. So that is condensation kind of reaction. And if we can say peptide bond breakdown, we will say it is hydrolysis, because water must be involved in order to break down the bond. So a peptide bond, a dipeptide contains two amino acid. Di means two amino acid together. So a dipeptide is two residues joined, two amino acids joined together. So also have peptide bonds are broken by, remember I told you, formation of the peptide bond is condensation. Breakage of the peptide bond is hydrolysis. So since when you're forming the bond, water is released. When you're breaking the bond, water must be incorporated to break the bond. And when water is incorporated to break a chemical bond, we call that kind of breaking hydrolysis. Use of water to break down bond or that cliff bond. That's what we call hydrolysis. So we have the bond between cysteine residues. the bond between cysteine residues is formed by, it's called the disulfide bond. It's called the disulfide bond. That bond will link two cysteine. We have, let's just say we are drawing the arrow group. The arrow group of cysteine has CH2SH. While the second cysteine you have CH2SH. So the bond between these two will liberate these two hydrogen. So hydrogen will be cancelled, and the bond will be formed between these two S. So it's called a disulfide bond. It's actually used to link two subunits. Can actually be used to link two subunits, or can actually be used to link proteins together to form a dipeptide. Not a dipeptide, sorry, a disulfide bond. So do not forget to click on the subscription button and the notification bell for more updates on videos on amino acid MCQs.